So this hallmark will sound pretty familiar, as we did cover it somewhat in defining biological immortality as a chapter. Regardless, we'll go into more in-depth into the actual science behind it as it pertains to humans, and not looking at nature so much. So let's have a tiny recap. Cellular senescence refers to a state in which cells lose their ability to divide and proliferate. One of the primary drivers of cellular senescence is the Hayflick limit, which is the maximum number of times a normal somatic cell will divide. Once this limit is reached, cells enter a state of replicative senescence and can no longer divide. Apart from reaching the Hayflick limit, cells can also become senescent due to various stresses, including DNA damage, oxidative stress, and inflammation. These stresses activate specific signaling pathways that trigger cellular senescence. Senescent cells often secrete a range of bioactive molecules, including pro-inflammatory cytokines, growth factors, and matrix remodeling enzymes. This collection of secreted factors is referred to as the senescent associated secretory phenotype, or SASP. The SASP can have both positive and negative effects, as it can influence tissue regeneration and repair, but can also contribute to chronic inflammation and tissue dysfunction. Accumulation of senescent cells in tissues over the years can contribute to the aging process. They can disrupt tissue homeostasis, impair regeneration, and promote inflammation. Cellular senescence has been associated with various age-related diseases, including atherosclerosis, osteoarthritis, and neurodegenerative disorders. Perhaps the most hot-button topic directly relating to cellular senescence is the use of senolytics. Senolytics are a class of drugs that have the potential to selectively target and eliminate senescent cells in the body. The two most common ones being used in clinical trials at the time of making this video are dasatinib and quercetin. Dasatinib is a medication that belongs to a class of drugs known as tyrosine kinase inhibitors. It is primarily used as a targeted therapy for certain types of cancer, particularly in the treatment of chronic myeloid leukemia and Philadelphia chromosome-positive acute lymphoblastic leukemia. The BCR-ABL fusion protein is formed when the two proteins, aptly named BCR and ABL, fuse together, creating an abnormal tyrosine kinase. This protein is responsible for the continuous activation of signaling pathways that promote growth and division in leukemia cells. Dasatinib specifically targets BCR-ABL and inhibits its activity. By blocking the activity of BCR-ABL, dasatinib disrupts the signaling pathways that would otherwise lead to uncontrolled cell growth and division. This inhibition of signaling cascades helps slow down and halt the progression of leukemia. Dasatinib can also trigger apoptosis. In cancer cells, this is often suppressed. Dasatinib can help restore the ability of these cells to undergo apoptosis, thereby reducing the leukemia cell population. Apoptosis can be triggered by various internal and external signals. Internal signals can include DNA damage or the presence of misfolded proteins, whilst external signals come from neighbouring cells or the environment. These signals activate specific pathways that initiate apoptosis. Caspases are a family of protease enzymes that play a central role in this process. Two main categories of caspases are involved: initiator and effector caspases. Initiator caspases include caspase 8 and 9 and are activated early in the process and serve to trigger the activation of effector caspases. Effector caspases include caspase 3, 6, and 7, which execute the dismantling of the cell. There are two pathways in which apoptosis occurs: the mitochondrial pathway, also known as intrinsic apoptosis, and the death receptor pathway, also known as extrinsic apoptosis. In the intrinsic pathway, internal stress signals leads to the release of pro-apoptotic factors from the mitochondria. These factors, such as cytochrome C activate caspase 9 and initiate the apoptotic cascade. In the extrinsic pathway, external signals bind to cell surface receptors known as DEF receptors. This binding triggers the activation of initiator caspases, particularly caspase 8. Once initiated, caspases cleave and activate other caspases, creating an amplification loop. This cascade of caspase activation leads to the proteolytic cleavage of numerous cellular substrates, ultimately resulting in cell death. Caspase activation leads to the cleavage of proteins involved in DNA repair and maintenance. This causes DNA fragmentation and chromatin condensation, leading to changes in the cell nucleus. Apoptotic cells undergo a series of structural changes, including cell shrinking, membrane blebbing, which is the formation of bubble-like protrusions, and the formation of apoptotic bodies. Cell remnants are recognized and engulfed by phagocytes, such as macrophages. This ensures the safe removal of apoptotic cells without causing inflammation. This process helps retain immunological tolerance by eliminating self-reactive immune cells and avoiding autoimmune responses. Apoptosis is a tightly regulated process that prevents damaged or unwanted cells from releasing harmful contents into the surrounding tissues, thereby maintaining tissue homeostasis. Because of its use inducing apoptosis in cancer cells, dasatinib is also being investigated as a senolytic, whereby it works by disrupting the pro-survival mechanisms that allow senescent cells to persist. Senescent cells often activate specific pro-survival signaling pathways that protect them from undergoing apoptosis. Dasatinib interferes with these pathways, making senescent cells more vulnerable to this process. This leads to the removal of these dysfunctional cells from the body. Because senescent cells secrete pro-inflammatory molecules as part of the senescence-associated secretory phenotype, eliminating these senescent cells helps reduce the levels of inflammatory molecules, thereby reducing chronic inflammation and age-related diseases. Quercetin is a natural flavonoid compound found in a variety of plant-based foods. Quercetin is a potent antioxidant, which means it can help neutralize harmful free radicals in the body. Free radicals are molecules with unpaired electrons that cause cellular damage and contribute to various health issues, including aging and chronic diseases. Quercetin's antioxidant properties can help protect cells from oxidative stress. As with dasatinib, it can affect signaling pathways of senescent cells, promoting apoptosis and thus having anti-inflammatory effects. Related to senolytics are xenomorphics. Xenomorphics encompass a wide range of agents that can modulate the phenotypes of senescent cells to those of young cells by interfering with xenoinflammation and senescence-associated secretory phenotype without inducing apoptosis as senolytics do. Xenoinflammation refers to chronic, sterile, low-grade and unresolved inflammation characterized upon aging. Nuclear factor kappa B is activated in aging and is a key transcription factor mediating inflammation. It has been linked to known regulators of the aging process, including insulin-like growth factor, sirtuins, mTOR, and DNA damage. A prominent xenomorphic compound is apigenin. One study showed apigenin to be a peptide inhibitor of the nuclear factor kappa B activating kinase, which reduced cellular senescence in vitro and in vivo and delayed the age-related symptoms and pathologies of progeroid mice. Both senolytics and xenomorphics can have antioxidant activity. Speaking of antioxidants, the other one that is usually talked about is resveratrol. Resveratrol is a natural polyphenol compound found in certain foods, particularly in the skin of red grapes. It can help neutralize harmful free radicals in the body. It does this by donating electrons to these radicals, thereby stabilizing them. In addition, it upregulates the expression and activity of endogenous antioxidant enzymes such as superoxide dismutase, catalase, and glutathione peroxidase. It can also chelate, which means to bind to, and it binds to metal ions such as iron and copper. These ions participate in chemical reactions that produce said free radicals, further reducing oxidative stress. In addition to their interactions with free radicals, resveratrol plays an anti-inflammatory role. It activates sirtuins, supports mitochondrial function, protects from DNA damage, promotes autophagy, and improves cardiovascular health. Resveratrol can be found naturally in red wine, but be careful not to drink too much, otherwise you may have liver problems instead to contend with.